Assalamualaikum warahmatullahi wabarakatuh. My name is Mentari Luthfika Dewi. I am one of the lecturer of Pharmacy Department, Bandung Islamic University. Today I want to present my research report at the third Science and Technology Research Symposium 2020. Firstly, let us say thanks to Research Institute and Community Service LPPM Bandung Islamic University for financially support on this research at the Young Lecturer Incentive Grant Program 2019. These presentations will be divided into 4 sections. Firstly is introduction, then method, result, and conclusions.
The title of this research is Computation Study of Phthalocyanine-Cyclodextrin combinations against Histone Deacetylases for Photodynamic Bladder Cancer Therapy. Our research teams are Mr. Taufik Muhammad Fakih, Mrs. Anggi Arumsari, Nurfadila Hazar, and Tanisa Magviro. All of us are from Department of Pharmacy, Faculty of Mathematics and Natural Science, Bandung Islamic University, Bandung, Indonesia. Cancer of the urinary bladder is a highly prevalent disease, and it's closely associated with substantial morbidity, mortality, and cost. It is the 9th most frequent malignancy and the 13th most common cause of death worldwide. Developed countries are nearly 3 times more vulnerable to bladder cancer than underdeveloped countries, with North America and several European, Western Asia, and Northern African countries exhibiting the highest age-standardized incidence. The American Cancer Society estimates in 2019 bladder cancer became one of the 10 leading cancer types in the United States, with estimated new bladder cancer cases about 61,700, and it caused 20,800 death cases. The annual incidence now number over 350,000 new cases worldwide, but it's estimated to approach 800,000 within the next 5 years, largely due to the aging of population and the increased prevalence of cigarette smoking. Urban living and diet low in fruit and vegetable are additionally linked to increased incidence of bladder cancer. There are some studies report that primary cultures of human cancer cells and various human tumor biopsy samples often identify a higher level of HDAC expression than normal tissue. Recent evidence suggests that clinical samples of patients with bladder cancer and tumor tissue from most models have shown significantly increased HDAC expressions compared to other healthy tissues. Thus, the HDAC inhibitions may be an effective option for treating bladder cancer. The decrease of histone acetylation by HDAC inhibitors, or we call it HDACI, leads to the modification of expression of several genes implicated in oncogenic properties of cancer cells. From top left to bottom right, HDACI can reduce angiogenesis and tumor growth, also improve treatment by inhibiting DNA repair. Also, it can induce cell cycle arrest and stimulate apoptosis. There are several HDAC inhibitors which are still used as potential chemotherapeutic agents for bladder cancer, as well as the backbone systemic chemotherapies. But most of these treatments will eventually develop chemoresistance. So, it's been urgent to develop some specific selective anti-cancer agent with minimal side effect and minimal chemoresistance. This is phthalocyanine. It's well known as an aromatic macrocyclic with excellent photophysical properties for use as photosensitizer in photodynamic therapy or PDT for cancer treatment. This therapy combines visible light, molecular oxygen, and photosensitizer capable of producing reactive oxygen species, or we call it ROS, such as single oxygens, which can induce cell death pathway that result in tumor tissue damage. The conjugation of phthalocyanines with biochemical motifs is very important in development of a promising photosensitizer because it can increase their solubility in water and can act carriers for phthalocyanines to deliver them to cancer cells. And this is cyclodextrin with alpha, beta, and gamma conformations. It's consisting of 6, 7, and 8 glucopyranose units that bond to the glycosyl linkage. Previous research reported that conjugation of photosensitizer with cyclodextrin molecules can increase amphibicity, biocompatibility, and availability on the surface of cancer cell membrane. So this research is to identify, evaluate, and to explore the mechanism of actions of combinations of phthalocyanines and cyclodextrin molecular structures on HDAC macromolecule by utilizing in silico molecular docking simulations. So this is the software and hardware that we use. And for the material, we use complex molecule zinc-phthalocyanines with alpha, beta, and gamma cyclodextrins. And for the target macromolecules, we use crystalline structure of HDAC in bladder cancer, which form a complex with pheronostat as a natural ligand. And for the method, firstly, we have to prepare HDAC macromolecules by removing water molecules and pheronostats and then adding polar hydrogen atoms and calculating Coleman's partial charge. And also we have to prepare phthalocyanines and cyclodextrin molecule complex with selected for the best conformation based on the total energy value. And for the simulations, we use PatchDock software. And the result of molecular docking simulations are then identified and compared based on the binding free energy value. Also, we have to explore and evaluate the molecular interactions that are formed by using BioVia Discovery Studio 2020 software. And this is the result. We have performed zinc-phthalocyanines with alpha, beta, and gamma cyclodextrin complexes in three-dimensional structure using PatchDock software. And the best results are selected based on the total energy value. As seen in Table 1, it can be predicted that these complex molecules can form a good affinity for histone deacetylases molecules. Based on the docking molecular result, as shown in Table 2, it estimated that 3 complexes molecule of phthalocyanines and cyclodextrin have a better affinity when compared to natural ligands. As we seen in Table 2, the value is more negative than the binding free energy of pheronostat, the natural ligands. As seen on 3D figure, it can be observed that overall 3 complexes of phthalocyanines with cyclodextrin have the same conformations as natural ligands and can occupy part of the active site of macromolecule histone deacetylases. From the binding free energy, we have known that the best affinity is shown by zinc-phthalocyanines and alpha-cyclodextrins complex. It can be seen in Table 3. It has many molecular interactions with HDAC macromolecules. It has 5 hydrogen bonds and 3 hydrophobic interactions. And the other complexes of phthalocyanines with beta and gamma cyclodextrins, it has 11 and 16 molecular interactions. Different with pheronostat, the natural ligands, it can only bind to 1 amino acid. Based on this phenomenon, it can be predicted that the complex molecule of phthalocyanines with cyclodextrins can bind more stable with HDAC macromolecules.
From this research, we can conclude the complex molecule of phthalocyanines and cyclodextrin is a potential photosensitizer candidate in the development of photodynamic therapy base for bladder cancer treatment.